Hi, my name is Mokshit Manuru, and I'm going to be presenting our project on mRNA processing in the event biotechnology and molecular biology. So first we need to discuss what proteins are. So proteins are a class of macromolecules that perform a diverse range of functions for the cell. They're very important for growth, development, and tissue repair. So that's why people are always like, eat a lot of protein, because they do all these amazing things for our body and they help us when we're hurt with the tissue repair. And an example of a protein is hemoglobin. Hemoglobin is a protein in your red blood cells that carries oxygen to your body's organs and tissues, and it also transports carbon dioxide from your organs and tissues back to your lungs. So they're very important and we need them to survive. So what is mRNA processing? So mRNA processing comes after transcription. When I said that, you're probably like, well, what is that? So let's talk about it. So we just talked about proteins. We need a way to make them. So how we make these proteins is through these processes called transcription and translation. So DNA contains the code to make proteins. However, DNA is in the nucleus and it can't always leave the nucleus when we need these proteins. So what transcription does is, using a strand of DNA, using this strand, it creates an RNA with complementary bases. So you see how it's TA, it just makes complementary bases. And it creates this RNA, and it creates a type of messenger RNA, and we use that message, and we create that in transcription. And then in translation, we use that message, and we code these different amino acids. And these different amino acids, they're put together to make polypeptides, which can be used to form the protein. So transcription is a process of making mRNA from DNA. And then after transcription, however, the mRNA is not quite ready for translation. So it needs to go through these extensive processing. That is why the mRNA made directly after transcription is known as pre-mRNA. And this process, note that only occurs in eukaryotes, because prokaryotes, they don't even have a nucleus. So they don't need all these processes to leave the nucleus and go to the cytoplasm where translation takes place. So what are the main processes involved in mRNA processing? So these processes are the 5-cap addition, which is right here. We're adding a 5-cap to our pre-mRNA, and then polyadenylation. You see this AAAAA, that's called a polyadenylation. Adding that is just polyadenylation. And then splicing. You see how we're cutting things? That's splicing. And we can think of this as this right here. I'm mature. So before these processes, think of these processes as the arrow. So before these processes, it's immature. And then after these processes, it's I'm mature. It's mature. So pre-mRNA, and then we turn into a mature RNA, which can be used in translation after these processes. So what is the 5-cap? What is the addition of 5-cap addition? What is the point of this? How does the mRNA know when to leave the nucleus, and when it does, how does it find the ribosome and attach to the proper place that it needs to be attached to? A signal at the end of the 5' end of a pre-mRNA helps with both of these jobs. The name of this signal is called a 5' cap. This 5' cap is a modified guanine nucleotide. So this is what the structure of this thing looks like, this 5' cap. So what is the function of this cap? The cap helps the mRNA strand bind to the ribosome during translation. So the ribosome is the site of protein synthesis. That's where we make our protein, and that's where translation occurs. And the 5' cap also helps with protection from exonucleases. Those are enzymes that degrade nucleic acids. And most of our RNA is made from nucleic acid, so not be very good if we were getting hurt from those things. So that's why we need this cap. So this is the 5' end, and we're just attaching this cap to this to protect it and find the proper site on the ribosome. Next, we're going to take a look at polyadenylation. What is this? This is a process in which a polyadenylate is added to the 3' end of the mRNA. So we can see here, AAAA, we're just adding a polyadenylate to this 3' end, and that's called polyadenylation. So this polyadenylate can have 80 to 250 adenosine residues, and it's very important because it also protects the mRNA, like the 5' cap, from degradation by exonucleases. And polyadenylation, one of its key functions, and why such a key process, is because in mRNA processing, it facilitates the release of the mRNA from the nucleus into the cytoplasm. So remember, in transcription, we're making this mRNA. That happens in the nucleus because DNA is located in the nucleus. And then we need to get to the cytoplasm where the ribosomes are. And polyadenylation helps with this. And eukaryotic cells, the transcription of polyadenylation signals indicates the termination of this process. The mRNA transcript is then cut off the RNA polymerase and then freed from the DNA. So, splicing. First, let's talk about this Human Genome Project. Before the Human Genome Project, scientists thought that there were about 100,000 genes, but we only actually have about 22,000. This can be explained due to a process called alternative splicing. Alternative splicing allows different RNAs to be produced from the same primary transcript. This depends on which part of the RNA segment are treated as exons and what are treated as introns. So first, to understand this, we need to understand the structure of the pre-mRNA. We'll take a look at this in this slide. So we have introns, which don't code for anything, and exons, which code for things. So based on what is used as exons in the pre-mRNA, we create different mRNAs. This final mature RNA that we use in translation, it can be different based on what we use as exons. So sometimes this part right here, that could actually be used as an exon, and this could be used as an intron. That could change the protein we make because a different mRNA is being used. And these are actually spliced out by these parts. These introns are being spliced out by S and RNPs within spliceosomes, which help up with this splicing process. This allows just the exons to leave the nucleus, and this is why the mRNA that leaves the nucleus is much shorter than the primary transcript. So let's take a look at this here, this alternative splicing process. So you see we have these exons, right? 1, 2, 3, 4, 5. Here, the exons, all of them are being used, and we make a protein A. And here, 3 is being skipped. It's just 1, 2, 4, and 5, and protein B is being created. And you see, with the same strand of mRNA, we can make different proteins because we're making different mRNAs because we're using different things as exons. So, Alzheimer's disease, and this is the real-life application part. What is Alzheimer's disease? So the online definition of Alzheimer's disease is that it is an irreversible progressive brain disorder that slowly destroys memory and thinking skills, and eventually the ability to carry out the simplest of tasks. So let's take a look at abnormal mRNA splicing, and not same thing, you know what I mean. And Alzheimer's disease, newly funded. NIA funded study, not newly funded because it is from 2018 December, but finds that changes in a process called RNA splicing interact with specific Alzheimer's genes and may contribute to functional deficits in the Alzheimer's brain. So, researchers from the Icahn School of Medicine at Mount Sinai and Columbia University studied brain tissue from more than 450 deceased older people in 2 NIH-funded studies. And they created a genome-wide map of splicing variations in the dorsolateral prefrontal cortex. The dorsolateral prefrontal cortex is typically associated with executive functions including working memory and selective attention. So, the analysis of the RNA sequence data showed that the brains with Alzheimer's disease had more alternatively spliced RNA messages than the brains without Alzheimer's disease. Those altered splicing events corresponded to 84 genes, including a number of newly discovered risk factors for Alzheimer's disease. The findings suggest that altered RNA splicing in the brain is a feature of Alzheimer's disease that is in some cases genetically driven. So, were you paying attention? Let's find out. Which of these is not a main RNA processing process? A. 5-cap addition. B. Polyadenylation. C. Single-cell genome editing. And D. Splicing. I'll give you a second. And if you pick C, you are correct. Single-cell genome editing is not a mRNA processing process. And these are the sources for this video. I hope you enjoyed, and that's the end.